My research now is focused on innate immunity and how aspects of innate immunity are regulated by phospholipids. And when we say innate immunity, we mean that aspect of immunity that occurs before you develop antibodies. And so most people have heard of antibodies, but the innate immune system does not require the presence of antibodies, and it's actually the first step that occurs in host defense against mostly pathogens. It's a really long history. As a graduate student, I was mostly interested in protein biochemistry and enzymology, and I did a lab rotation that was focused in a laboratory that was focused on lipids, particularly lipid metabolism and enzymology with respect to the biosynthesis of lipids and membranes that are involved in constructing lipids. From there, during my tenure as a graduate student, I moved into an area of pulmonary surfactant lipids. Pulmonary surfactant is a lipid and protein complex that lines the gas exchange areas of the lung. And I became interested in the enzymology of how the major lipid in pulmonary surfactant, which has a long name, dipalmitoyl phosphatidylcholine, is made. Because we knew that the de novo pathways for synthesizing lipids generally did not make molecules like dipalmitoyl phosphatidylcholine. And so part of our work was able to elucidate that the way this unique sort of phospholipid is made is by first making a precursor lipid and then partially degrading it and then synthetically using a certain type of reaction called an acyl transferase reaction to make this dipalmitoyl phosphatidylcholine. I was attracted to this particular area because at that time the major cause of neonatal mortality, especially in premature infants, was insufficient production of pulmonary surfactant, and in particular this lipid called dipalmitoyl phosphatidylcholine. So that's how I spent my graduate education, was really doing lipid biochemistry and lipid enzymology. And then from there I did a postdoc in Boston with Gene Kennedy, who in some respects is considered sort of the great-grandfather of all lipid biochemistry in the US. And I really became interested in what was a major gap in our understanding of how membranes are made, and particularly how the lipid components of cell membranes are moved from their site of synthesis, which is primarily the endoplasmic reticulum, to the other membranes that characterize eukaryotic organisms. So for most of my career, my independent career, I focused on these issues of lipid transport with regard to membrane biogenesis. And about 10 years ago, I returned to this discipline of pulmonary surfactant and the lipids of pulmonary surfactant because we, quite by accident, discovered that some of these minor lipids in pulmonary surfactant, they go by the name of phosphatidylglycerol and phosphatidylinositol, were very, very potent regulators of inflammatory processes in cells. So I would say really over the last decade I've transitioned from studying membrane biogenesis per se to the regulation of innate immune processes by two phospholipids, phosphatidylglycerols and phosphatidylinositols. So what our research has taught us is that inflammatory pathways that are elicited in the innate immune system by microbial products such as endotoxin or LPS, or double-stranded RNA or double-stranded DNA, or lipopeptide fragments can be very remarkably suppressed by these two lipids that exist in pulmonary surfactant. So this was a surprising finding at the time, and our work in this area has continued, and I guess through another sort of serendipitous aspect of my career, where we were studying these lipids and how they suppress inflammatory processes elicited through these innate immune receptors known as toll-like receptors. We started studying inflammation as a consequence of viral infection. And we focused on two viruses, initially just on respiratory syncytial virus. Okay, and our interest in respiratory syncytial virus came from a number of directions, but probably the most prominent direction that it came from is the fact that this is a virus that infects all children before the age of 2, and there's no vaccine for this virus. And this virus also is the chief cause of newborn hospitalization in the US. And worldwide, it's a much even more significant problem because it's a primary cause of newborn mortality. So the numbers for newborn mortality are just astonishing. So as many children die from respiratory syncytial virus infection as do from malaria worldwide. So this is in the hundreds of millions. So in our studies, what we've begun to focus on is how the inflammation that's elicited by the viruses is inhibited by these surfactant lipids, the phosphatidylglycerols and phosphatidylinosytols. And quite surprisingly, what we found is not only is the inflammation elicited by the viruses inhibited, but it turns out that viral infection is inhibited by these lipids. So in a typical experiment, we may take mice and introduce respiratory syncytial virus in their nasal passages, and they will become infected. But if we introduce the respiratory syncytial virus along with some of these pulmonary surfactant lipids, we find that the infection is almost completely prevented. So again, this is a virus that where there's a high degree of worldwide mortality. Its primary cause of hospitalization in infants in the US, and there's no vaccine. And now we find that we can just apply some of these lipids, and we've not done the experiments in humans yet, but when we do the experiments in mice, we can give the mice these lipids down their airways, and we can protect the mice from viral infection. And our success with respiratory syncytial virus led us to ask questions about other respiratory viruses. And so here we've begun to focus on influenza A viruses. So the influenza A viruses, generally we think that in general it's thought that they're not such a big problem because we have annual vaccines. But it turns out that if you look at the history of vaccines for influenza virus, especially this year in the 2017-2018 flu season, the viruses are marginally effective at best. And over the last 10 or 11 years, the overall efficacy of influenza vaccines has only been 41%, which means that if you get vaccinated, there's a 59% chance that you'll not be protected from the virus. So we need new ways to treat influenza viruses. And so what we found is that both of these lipids, phosphatidylglycerol and phosphatidylinosytol, bind to viruses and prevent viruses from engaging in the very first step of infection, which is binding to cell surfaces. And so now our work is really directed towards understanding at the very finest structural level how these lipids interact with viral surface proteins and prevent the viruses from binding to cells and ultimately infecting cells. And I should also say that we've looked at several species of influenza virus. That includes mouse-adapted viruses that we use for our mouse experiments, also H3N2 influenza viruses, which were the prominent cause of infectious problems this year. And we've looked at pandemic H1N1 virus, which emerged several years ago as a virus for which there seemed to be very little, if any, pre-existing immunity. So anyway, with these three viruses, we find that the effects of both lipids are the same, that they prevent the viruses from attaching to cell surfaces and infecting cells. So our long-term hope is that we can use these lipids in a supplementary way to vaccination to prevent viral infection. And we think there also may be application of these lipids if people are not vaccinated, that may be to prevent them from being infected until they can get vaccinated. And also with the sort of the efficacy rate of the vaccinations being on the order of 50% or lower, this would afford additional protection for individuals who don't mount a sufficient immune response elicited by the vaccines. And I guess the final thing that I'd like to say about this is the 1917-1918 flu pandemic basically killed 5% of the world population. And many epidemiologists feel that this is going to happen again. It's not a question of if it will happen, but when it will happen. And so I think all of the types of antivirals that we can mount to try and prevent large-scale mortality worldwide if an extremely virulent strain of influenza arises will be a problem, but we think maybe the lipids that we work with and preparations of the lipids that we work with will maybe protect a large portion of the population from the lethal effects of a highly virulent virus.